Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. This is a very special episode. We talked to Dr. Steve Ferguson from the University of Ottawa about the history of my favorite GPCR meeting. You know which one? The joint Great Lakes GPCR retreat and the Club des récepteurs à 7 domaines transmembranaires du Québec. Mark your calendars for the 21st edition of this scientifically rich yet intimate meeting where I will also be present. If you are already registered or plan on coming, I look forward to seeing you and invite you to stop by my poster. This meeting is held between May 12th to 14th at Niagara-on-the-Lake in Canada. Visit this episode's page on drgpcr.com/podcast to learn more and register. There is still time to join us. And now, let's dive in. Hello, everyone. This is Yamina with Dr. GPCR. I'm very excited today to have Dr. Steve Ferguson with me. Hi, Steve.
Hi, how are you doing?
I'm great. I'm so glad I have you on today. And before we hit record, full disclosure, we just recorded a mini episode around the GPCR retreat of this year, and we've been talking a little bit. I'm super excited to be talking to you about your work, your career, and let's hit it with the first question that I typically ask is, how did you end up studying GPCRs?
Well, I didn't originally want to study GPCRs, so I did my Ph.D. on the choline transporter in the brain, which is what transports choline into neurons to make ECH. Marc Caron came to give a departmental lecture. I really liked what he was doing with the dopamine transporter. He invited me to come give a seminar. So on the way back from my honeymoon, my wife and I stopped in at Duke, and the lovely Duke Medical Center in at the time. And Mark was kind enough to offer me a position as a postdoc. And so I came back 6 months later when I finished my PhD, ready to work on doing dopamine transporter chimeras, trying to get an idea of the structure function. And I was told I was going to work on beta-2 adrenergic receptor internalization and the role of phosphorylation. So that turned into working on beta-arrestin as an acidic molecule, and the studies with Jay Zhang and Stefan Laporte and Larry Barrick, using, Larry introduced the idea of fluorescent proteins and studying GPCR signaling. It's turned into a lot of the work Michel Bouvier and Stefan have pushed with the biosensors. And so, I discovered that GPCRs were more fun because they're more dynamic. Transporters they're actually kind of boring. But I was lucky that Mark pushed me in the direction he did.
And was that the first time that you heard about GPCRs? Is there a time that you remember going back in your training where you kind of heard about GPCRs and thought, well, might be interesting, but then moved on to working on transporters?
Well, I probably should have heard about GPCRs as an undergrad because, unfortunately, I was a terrible student. I never showed up for class. I don't even know what my second year biochemistry professor looks like. I wrote the bubble test and somehow managed to get a 55 because my roommate made me get out of bed and go. But as a graduate student, I became quite aware of them because Bob Lefkowitz came as a visiting professor for one of the more prestigious McGill visiting professorships, which I think he's done again. And he gave 2 talks, mostly focused on Brian Kakoka's work, and I thought that was extraordinarily interesting. So
that was the first time, but it didn't catch your imagination at that time until you went into.
Well, I didn't really know what I wanted to do. When I finished my undergrad, my father looked at me and says, "What are you going to do?" I said, "Well, maybe I'll become a technician." He said, "Why would you do that? It's a dead-end job." I'm like, "Oh, okay." I had an interest in pharmacology for reasons I won't disclose. So, I've been working at the Montreal Neurological Institute with Tony Hakim, doing positron emission tomography, and I sort of got sold off to Brian Collier to do a project we'd come up with to try and use an enantiomers of choline to selectively image cholinergic neurons. I had an interest in Alzheimer's disease. I was doing imaging of Alzheimer's patients, looking at changes in blood flow in the somatocentric cortex as they counted backwards from 7. Amazingly, you can see a huge impairment in somebody who has dementia compared to somebody who's not dementia. So it was a project that I was interested in. it didn't work. Turned out to be one of the most boring projects one could ever have done, but it did teach me how to do science. And Brian Collier was, an excellent mentor. I thought about going for a postdoc at Western because my wife was a teacher in London, and I remember Brian coming up to me one day. He used to like to give a big sniff, and he said, "Fergus, University of Western Ontario is not Duke University Medical Center." I went, okay, I get the point. Please don't tell me I'm stupid again.
That was interesting because there was no data associated to that statement. It was a blunt statement, and this was it.
Well, the Howard Hughes Medical Institute at Duke and Department of Physiology at Western, there's no comparison.
Yes. Definitely. You mentioned that going to give a seminar in Marc Caron's lab, you were coming back. It was a stop on your honeymoon. May I ask, where were you on your honeymoon?
Oh, our honeymoon almost caused a divorce, actually. My wife got a phone call and accepted one of these, come down and look at a timeshare condo. You can go for a cruise for free. She thought that was a great way of having an inexpensive honeymoon. So we drove all the way down to her father's condo in Florida for the week. Went to these condos where they tried to do the hard sell on you. Went out for a cruise and then drove back up to North Carolina. And, was not used to the heat back then. Late July in Florida is really miserable. It's kind of like January in Ottawa, but the opposite.
I can only imagine. I mean, I know that January in Montreal is definitely no fun.
Actually, Montreal's slightly warmer than Ottawa.
Interesting.
Yeah. So I lived 10 years in Montreal. I lived 7 years here. I was much warmer in Montreal. Maybe because I was a younger man. I don't know.
I don't know, but I haven't lived in Ottawa, and I've been in Ottawa, but I don't think it was in the winter time. So I can only speak up to the cold in Montreal. As let's go a little bit further back, and then we can move forward. As a teenager, did you know what you wanted to become? Was there anything that you were interested in more? Sometimes people say chemistry, others say astronomy, others say, becoming a doctor.
Well, I'm very thankful that Ontario had grade 13 back in those days, because until grade 12, I was barely passing high school. And in grade 12, I decided that I wanted to go to university, so I actually started paying attention. The only thing that I couldn't do effectively was learn French. Now, that was mostly my fault, but it also was partly the fault of my grade 12 French teacher who spoke French with a thick Glaswegian accent. And his only claim to fame of being a French teacher was he served in France during the Second World War in the RAF. So I ended up dropping that. But, I found history easy. I found maths and physics and chemistry extraordinarily easy. Biology was a little bit more difficult. Geography is probably what I should have done. Probably should have become a geologist. But I had a father who was pushing me hard to go into medicine. So
I know the sentiment. I definitely know.
It took me a while before I pushed back with my mother's help. So
well,
it's hard to grow up in the house with a world famous neurosurgeon.
There's some pressure there.
Yeah.
Who
would always remind me that he finished his Ph.D. in 2 years.
Tough. Yeah. Well, but we're all different. My father is not an MD, but we have in the family many doctors with different specialties. And there was that pressure of saying, well, you must be an MD. And I have to say that I got rejected twice from the University of Montreal, and reading the rejection letter gave me this sense of relief, thinking, oh, this was not for me. And I'm just so glad that I didn't get in.
Well, I never told my father, and he'll never know because I lost him 10 years ago, but I never submitted my applications for medical school.
Okay.
I just told him I didn't get in.
Okay. That's one way to do it. Do you think you would have gotten in?
Based on my MCATs, probably not. As I said, I was a lousy student. The parts that were hard, where you had to think, look at data and explain it. Yeah, I did really well. But remembering first year biology and plankton and all that sort of garbage that they wanted you to spew back. Yeah.
Yeah. That's a different profession there. Definitely.
And he didn't do himself any favors taking me in to watch him do a neurosurgical case. I found the opening of the skull the most barbaric thing I've ever seen in my life. Once they started doing the neurosurgery, it was beautiful and delicate and eloquent. But getting in there, yeah, it's like cracking a walnut. It's really not. Anyway.
Yeah, I've never seen it live. I've only seen it on TV, and I sometimes cannot watch either.
Well, I was doing, as an undergraduate, I was studying behavioral ecology because I really liked the outdoors. And in second year, I decided it was for the birds because they're putting mathematical models to animal behavior. Anybody who's had a cat or a dog knows that animals don't behave according to an equation. They have spontaneous behavior just like we do. And I stayed in Montreal for the summer because of a girlfriend and couldn't find a job. Told my mother I was working in a flower shop. I'm a terrible liar. So she knew I didn't have a job. And it'd been several days without eating. And I finally got phone calls from McGill University that the government had opened up its summer training programs. And I qualified for 3 jobs at the Montreal Neurological Institute. And so I went and interviewed with a neurosurgeon who had no idea what my background was. And so it was to do this positron emission tomography project. So Ronald Kruger was a fabulous guy. Asked me, so do you know any computer science? I said, yeah, I took a course. I said, how'd you do? I failed. I said, okay. Do you know any statistics? I said, yeah, I took a course. I said, how'd you do? Well, I didn't fail, but I had to take it again. And so he took me on as a summer student over a number of medical students because he liked the fact that I was honest with him. And, I learned a lot from Rod. My first day in the lab, I was put in an office with a neurosurgical resident that was scraping away at the eye sockets of the guy's head that he'd frozen at minus 80 degrees. And I was thinking to myself, what in the hell I got myself into? But once I got into the positron emission tomography unit and worked with the nurses and the physicists and Tony Hakeem, who was spectacular, here in Ottawa as well, which is kind of nice. It changed my perspective, and I moved from behavioral ecology into neurobiology. So that's really sort of what first set me down the path that I was going. I had a grandfather who had Alzheimer's disease, and so I had an interest in Alzheimer's. Because when I was a young kid, I'd get a call from my grandmother, and I'd go up my bicycle and I'd find him because he'd gotten lost in the walk again. He'd go, oh, Steve, how you doing? Hi, Pa. Should we walk home? Go, yeah, sure. He was a gentleman's gentleman. Certainly very different than myself.
So you're in Marc's lab, Marc Caron's lab. You're starting to work on beta-arrestin and beta-2 adrenergic receptor signaling. What happened next? What are the most relevant discoveries that you made during your postdoc?
The biggest discovery I made was I don't know how to read. So I was looking at a Drosophila paper by Charles Zucker where he made 12,000 different mutations in visual arrestin in Drosophila. And he had these 4 mutants that were non-functional. And what I didn't realize is that there was an asterisk beside the one that I decided to make the mutation in beta-arrestin. And that asterisk was that it didn't express. So I missed that. So I made the mutation. Had been looking at the role of arrestin in desensitization. Was finding it was taking 20 or 30 minutes before it had any effect. So I decided to overexpress beta-arrestin in this V53D mutant. And it turned out the V53D mutant antagonized internalization. It resulted in a science paper, put together the data in 4 weeks. Bob Lefkowitz suggested a very good experiment looking at the synergism between GRK phosphorylation and arrestin. Took several months to convince Marc that we should send it to Science. And we got the paper back 4 weeks later, and the editor basically said, cut 400 words and we'll accept it. I've taken the liberty to cut 125. So 2 hours later, it was back in the FedEx box going to science. I learned later that publishing in high impact journals was much more difficult than that.
That was, I think that was a very separate and unique situation there. But
I think the greatest pleasure was working with Jay Zhang, who was a student in the lab who did some really cool work. She probably did some of the first bias signaling that was ever done, but we didn't realize what it was at the time. With Larry Barrick putting GFP on beta-2 adrenergic receptors. I thought he was out of his mind, but it turns out the receptor was better coupled. And then the first experiment looking at beta-arrestin translocate. Who would have thought the whole protein moved en masse to the cell surface if you expressed it at low enough levels to see the mass action? And my biggest regret is Duke didn't patent it properly, so none of us are independently wealthy yet. But
you mentioned that sending the manuscript was happening in a box and FedExed.
Yeah, we used to spend hours standing by the fax machine, hoping that we'd get our reviews back. It was very much different back then. You cut your gels out. You walked across the Duke campus to photography with everything mocked up on a piece of paper. They'd take pictures. You go back and get them. You then cut those out to put onto your graphs. It was a different world. You couldn't Photoshop anything. There was no falsifying a blot because you've changed the dynamic range of the pixels in the back. So you really had to learn how to get an autoradiograph that accurately represented a reasonable range of intensity, what you're seeing in your blot.
Wow. And now that I'm thinking that more and more journals request the original data, as it looked like, but as you mentioned, there is no way of falsifying that. You can't do anything because the result is the result. And
there were ways. If you're really dishonest, you could expose the autoradiograph to various radioactive substances.
But you shouldn't. I mean,
there was a big scandal in a major university in the northern United States regarding that. But yeah, it's pretty difficult to fake.
Yeah, and you shouldn't. But I think it also perhaps had that charm of having to go through all of these steps, and I think it made that fax coming in with the reviews and potentially with the acceptance letter even more appreciated.
Mm-hmm. Seemed more real back then.
Do you still get that excited when you get an email with reviews or with a decision on a paper as you were when you were waiting by the fax machine?
Well, it's a lot different now because it's more international. So the last paper I got the reviews back for last week, the reviews came at 2:00 in the morning. They turned out to be very positive, which was nice. But what people are expecting from papers nowadays, I don't think is necessarily reasonable. So you're seeing students having to put what should have been 3 papers worth of data into 1 paper with 25 supplementary figures that I guarantee you will be lost in the mist of the internet. It's going to disappear. So I don't think that's quite right. There are journals now that are saying we will only publish what is going in the paper. So you need to put together a story in such a way that it is a finite, concise story. Mm-hmm. I like that idea. I'm not a big believer in supplemental data. I don't have a difficulty with supplemental data where you have to show each of your immunoblots so that the reviewer can eyeball each of your blots and decide whether or not your analysis is actually correct.
Of course.
I also like the fact that some journals are now requiring scatter plots so you can see where each of your data points fall on the bar. And I like journals like British Journal of Pharmacology that won't accept anything less than an N of 5.
Wow. Interesting.
Because some of the journals figured out that you can't really have much in the way of statistical power if you're just doing an N of 3 and comparing 5 bars. Can't be done.
Wow. Times change. So we're far away from waiting by the fax machine for reviews.
Yeah. Nowadays, you've got to explain to students that, you can't get an error bar within a tube. Even if GraphPad Prism will give it to you, statistically, it's not possible.
Definitely not. Definitely not. So you mentioned your time in Marc Caron's lab. What happened next? Did you always know, well, always, I mean, while during your time in Marc's lab, did you think about what was going to be the next step for you? Did you think, oh, I'm going to be a professor, or did you have any plan at all?
I realized when I got to Marc's lab that I needed to work really hard because I'd taken my wife away from a very good paying teaching job. And so I felt a responsibility to her to succeed. So, put in long hours. We worked in a group of 5 postdocs, 4 postdocs and a student. And so the productivity was fairly high. Many of the postdocs in Marc and Bob's labs, which were very intermingled at the time, had the sole goal of going to industry. I came from, a family of academic clinicians. My father, as I said, was an MD, Ph.D. So I never really thought much about going to industry. I always decided I wanted to become a professor. I was lucky enough to be in a position at that time where there was 2 job openings in Canada, and I managed to get 2 of them. So I had a choice. So one just happened to be in the place that I grew up, which was London, Ontario. So
What was the other one?
Here in Ottawa with Tony and King.
Okay. Wow. So
when I interviewed the second time, Tony was kind enough to say, "Well, it's really nice that you started your career with me, and I'm going to finish mine with you." He's the elder statesman of neuroscience here in Ottawa. He really is a spectacular individual. So, both job offers were equivalent. I asked the director of the Robarts Research Institute what happens with my startup. He said, "Your startup." I asked Ottawa what happens to my startup if I get an RC grant. They said, "Well, we'll take your startup back." I went, my wife's family, my family, are all in Ontario, and I get to keep my startup if I get a grant. That was an easy decision.
Wow, that's amazing. Can I ask about
the startup? In Ottawa anyway, so.
Yeah. Can I ask about the startup?
What would you like to know?
What is it? What was the idea behind it? Anything you can share with us? The reason I'm asking, before you answer, the reason I'm asking is because oftentimes trainees, and I was one of them, when we're in the lab, we're so very much focused on the experiments, on the day-to-day, that sometimes we don't think about, okay, what's next and what are the possibilities? And I think showing that as a professor, you can build a startup, you can do many things, and then you can use the science, but also the scientific process to do good and to do great work in different contexts, is really important to highlight. Yeah,
so the startup was really sort of a new concept then. Before that, it was maybe $20,000-$30,000 at best. But now there was beginning to be some competition to get people back to the country. So I think my startup was $150,000 for research and $100,000 for equipment, which was pretty generous back then if you consider it was 25 years ago. Startup packages nowadays range from $100,000 to $2 million, depending on how badly the university wants somebody, right?
Yeah.
Now, the idea of giving the startup is really to get a young investigator going so that they can hire in a technician, students, if they're lucky, a postdoc, and generate new data that they can get independent grants with. So, it's invaluable, to be quite honest. The advantage of Canada over the United States, although our grants aren't as big as the Americans, is we don't have to pay for our own salaries. Salaries are guaranteed by the university in this country. I think that would revolutionize American science if that were the case in the United States.
It would make a difference. All right, getting back to science, and I ask this from everyone: what is your favorite GPCR or favorite GPCR signaling molecule? You get to pick.
At one point, it was beta-arrestin. Now, I guess it would have to be metabotropic glutamate receptor 5. Maybe a little bit M1 muscarinic receptors. At the time, the CRF receptor was a big interest in one. But any receptor that is involved in regulation of brain function is of interest to me. Sort of lost interest in the cardiovascular system.
You mentioned before we hit record that you do work with animal models. What are some of the current projects that you've been working on, and which ones do you find the most exciting? Obviously, all of them are exciting because you're working on them, but if you had to rank them.
Yeah, so I have to admit, moving into animal models was the most terrifying thing I did in my career. You know, the one thing you have to do as a scientist, and I hope younger people would remember this, is you've got to have faith in doing new things and that it'll work out, whether it's, moving to doing siRNA, to doing CRISPR-Cas9, to moving into most models disease. You've got to have faith that you will generate some data at the end of the day that you can interpret. So one of my favorite mouse models, the first one that really excited me was the first mouse model we made. We crossed mutant Q111 Huntington knockout mice with mGlu5 knockout mice. And what we found was we completely lost any of the Huntington aggregates in the brain. And I showed this data to a couple of Huntington biologists at the H.D. It was a company called HDMI, and they were absolutely stunned by it. So we were able to follow that up using a mGlu5 selective NAM and show that, again, we could improve the behavior of the mice as well as reduce the pathology. I went to Roche to try and pitch that because they had a drug that they'd already used in Fragile X syndrome and major depressive disorder, which was safe, so could potentially be repurposed. I was unaware that they had bought the rights to the siRNAs that were being used to treat Huntington's to knock down mutant Huntington expression. So they were closing their mGlu5 program, so they didn't have much of an interest. I'm not convinced that mGlu5 selective names are necessarily going to work for treating Huntington's or Alzheimer's. In a mouse model, they work very nicely in both. I think the most exciting finding and the most surprising finding of my entire career was the work that Cal and I did together, where we looked at both male and female Alzheimer mice that have been treated with, again, CTEP, the mGlu5 selective NAM. And what we found was the male mice responded to treatment. Not only did they show improvements in cognitive behavior, reductions in the amount of A beta pathology, and changes in the GSK3 beta signaling pathway. Female mice, if anything, got worse. Their A beta levels went up, and the GSK pathway was irrelevant. And so, everybody said, well, that's got to be because of sex hormones. So a student visiting from Brazil asked her to do cell cultures from male and female embryos so that we could remove the confounding factor of estrogen and testosterone in the media. And she found exactly the same differences. So at which point I asked another postdoc in the lab who'd come from Mario Tiberi's lab and was a very good receptor ligand biologist to see whether or not, because we and others had shown that there's actually a complex that forms between mGlu5, A beta amyloid, and cellular prion protein that allows A beta to signal through mGlu5, as well as prion protein to signal through mGlu5. So I asked her to look at binding of tritiated MPEP to both the male and the female receptor and see, what affinity does A beta have for the receptor? Because I've always been asked, why do you choose the concentrations of A beta you use? Well, that's what people say to use. And what we found was that A beta 42 displaced tritiated MPEP with very, very high affinity in male mice, but was utterly unable to displace binding to female mice. And with clinical colleagues at the University of Ottawa, we managed to get our hands on some control cortical and hippocampal tissue from patients and show that the exact same thing is true in humans. There's no binding of A beta to receptor. So, what is my holy grail now? Is to try to figure out why. The structure of the receptor is the same. Why would female tissue in pharmacology be so different for mGluR5? We've done a lot of proteomic type approaches. We have a candidate that I'm not going to mention, but it's a difficult question to look at fundamental biophysical differences in receptor signaling between a male and female mouse, where you're not expecting there should be differences in binding. We've treated mice with estrogen inhibitors, testosterone inhibitors. There's a whole literature out there from a lab in the US suggesting that estrogen receptors can bind directly to mGluR5. So that was a possibility. It didn't seem to work out for us. So, it was a big disappointment to me that this drug wasn't working in females, right? Because I lost my mother-in-law to Alzheimer's just last year, sort of hoping that we'd have something for her. So we decided to move on to the M1 muscarinic receptors because there's been a fair number of studies from the Lindsay lab and Jeff Kahn's lab showing that you could use mGluR1 selective PAMs and get some positive effects. And what we found was, in fact, the M1 PAMs work very nicely in females. So it provides at least an alternative target. So, as Cal had talked about in his podcast, that the idea that there may be sex-selective drug treatments is a real possibility. I don't know whether drug companies segregate their patient data between males and females or what they do, right? It'd be interesting to know the answer to that.
Yeah, I think this would be the right time to think about it and do it, because sometimes just by watching TV, sometimes there's ads for medications, and the sentence that hit me is that some of these are for people assigned male at birth and not for people assigned female at birth. So my guess is, depending on what the disease is, when looking at it, there might be a possibility, but it sounds like and I remember it from my conversation with Khaled about this. It's a very interesting story, and you were talking about how is that even possible when you remove the hormone component and it's still different from the mGluR5?
We were asked to overoptimize the mice and castrate the mice and see what that did to mGluR5 signaling. But what we found as we aged those mice, our mortality rate in the Alzheimer's mice was about 80%. So we never ended up with enough mice to do the experiment. Whereas our control mice, the mortality rate was 0. So there's something to do with the inflammation that was associated with the surgery that the Alzheimer's mice just didn't tolerate. So it makes one think about, what is the role of immune cells in the brain in the development of these diseases? And could there be something different between immune cells in females versus males? There's certainly evidence from the pain field that the mechanisms by which women discern pain and men discern pain involve completely different enzymes that end up with the same biological response. So, certain drugs are not recommended for certain ethnic groups because of preponderances of single nucleotide polymorphisms in the patients. In this case, we're not looking at a receptor with polymorphisms. We have looked at polymorphic mGluR1 receptors that have been associated with cancers. And some of those polymorphisms are very interesting parts of the receptor, such as the ligand binding domain, the Homer binding domain, and the G protein binding domain. So if I could do one thing before I finish my career, it would be trying to get an idea of why mGluR5 is different pharmacologically in a male versus a female mouse.
I think that answer might be very interesting, and anyone listening out there, if they have any ideas, they'll know where to find you. And if there's any companies that might be interested in exploring these differences and harnessing the potential of targeting mGluR5.
We did have one company that was interested at one point, but they got a new CFO as they're taking their mGluR5 antagonist into phase 3 trials. And he shut down the collaboration because he had no tolerance for any negative data coming out of any study to do with the drug. He didn't want to see a single dead mouse for any reason.
Oh
So
goodness.
it was a small company. It was a small company that didn't have a whole lot of money. So the rationale in my mind was perfectly legitimate.
Reasonable. Yeah. Yeah. So, but I feel like not necessarily in this context, but in general in science, negative data is also very good you have to know what doesn't work.
Yeah, and sometimes negative data points you in the right direction. It was negative data that pointed me in the right direction with figuring out that beta-arrestin was an endocytic molecule.
Yeah, exactly. What's next? What other projects are you working on? You mentioned the muscarinic M1 receptor, the mGluR5. Are there any other receptors that you're currently working on in your lab?
I've gone back to my first love, transporters.
Full circle.
Hmm?
I said full circle now.
Full circle, but looking in the context of Huntington's disease and in the glutamate, vesicular glutamate transporter VMAT3, which is expressed in a very limited number of neurons in the striatum and other areas of the brain. And I saw a presentation by Cellular at McGill University when he came to visit us in Ottawa. It sort of struck me the phenotypes that he was describing with these VMAT knockout mice look very much like what we got when we knocked out mGluR5 or antagonized it. So in collaboration with Cellular, we've actually crossed the VMAT3 mice with the Huntington knock-in mice. Mm-hmm. And what we're seeing, surprisingly, and this was a real Hail Mary, is that we're seeing a significant improvement in motor function and cognitive behavior in the mice. And hopefully in the next few months, we'll know what's happening to the pathology in the brains.
Sounds exciting. And it's funny because you're as I mentioned, full circle to transporters, full circle back to Ottawa.
We're back to working with a protein called optineurin that my first postdoc, Peter Alberg, pulled out in the U2 hybrid screen back in 1998. There was nothing known about it at the time. It was an unknown protein. There's now about 400 papers on optineurin, ever since it was described as being responsible for about 17% of open-angle glaucomas due to single nucleotide polymorphisms. Now they found polymorphisms in the protein that are associated with ALS, with Alzheimer's, and the proteins found in Huntington occlusions. And we showed back in 2005 that in fact optineurin worked with mutant Huntington to regulate mGluR5 signaling. So we're now investigating, what does optineurin contribute to disease in Alzheimer's and Huntington's mice.
Wow, phenomenal. And it always goes back to GPCRs. And this is another question that I typically ask my guests. And I know the answer to the question is yes, but I'd like a little bit of a more elaborate question. And do you think GPCRs are still good drug targets?
I think yes and no. It depends on the disease. I think it's already been proven that they're good drug targets. Certainly they treat cardiovascular disease very well. Glucagon receptors in diabetes. It's been very successful. For the treatment of a disease like Huntington's, which presents itself fairly in patients at a fairly young age, I think there may be a hope for treating that disease and slowing the progression at least. I think there's a hope, and I'm going to keep things sort of focused on neurodegenerative disease. I think there's a hope for treating early onset Alzheimer's, which is a very rapidly progressive and devastating disease that affects people as early as 40 or 50. For late onset sporadic Alzheimer's disease, I don't know. I think in this day and age, if you make it to 80, you're doing pretty well. So the window of opportunity to make things better has shrunk dramatically. So I used to be optimistic, but I'm unfortunately less optimistic than I was. One of my close friends runs the Alzheimer's clinic at Western, and his comment is, the only thing I am is the most hated doctor in London because all I do is take away people who have demented licenses. I really don't like that.
Wow. What type of molecules would we be looking at in the context of neurodegenerative diseases targeting GPCRs? Which GPCRs should we be thinking about in the context of these diseases, and what do we want to modulate if we know what to do?
Well, for cognition, certainly receptors that are involved in cognition. There's a large number of receptors that are. M1 muscarinic receptors are an excellent example. When I was growing up as a graduate student, it was always about the cholinergic hypothesis. And then they tried using muscarinic antagonists, and the negative side effects were so severe that there was no clinical benefit whatsoever. But I think with the advent of allosterism and the ability to target outside of the ligand binding domain for ACH, which is almost identical in each of the 5 subtypes, people like Arthur Christopoulos and Patrick Sexton have been able to separate the pharmacology of M1 through M5 receptors, and Craig Lindsay and Jeff Caron are certainly engaged in quite an excellent drug discovery program, inventing new derivatives based on some of their first hits against the M1 muscarinic receptors that I think have huge potential for the treatment of cognition.
Fantastic. All right, last section, inspiring young scientists. We talked about it a little bit, about making the choice of being in a lab and then thinking about working hard, as well as you mentioned Marc Caron saying, well, have a good evening on Friday night and see you tomorrow, and he meant it. What would be your advice for junior scientists aspiring to contribute to the field?
So I guess you've got to decide what it is you want to do, whether you want to do industry or if you want to do academia. Industry in Canada is a little bit more difficult than the United States. My son's godfather lives across the street from former Prime Minister Stephen Harper. He and I were out barbecuing a few years ago in the summer, and Harper came across the street to talk to us, and he asked me what I did. I said, I'm a scientist. He says, oh, you're in industry? I said, no, I'm in academia. He said, why are you in academia? Money's in industry. And I looked him in the eye and said, there's not much industry left here in Canada, sorry. I didn't tell him that I thought it was his fault, but
that was going to be my next comment as well.
No, I managed to hold my tongue for once. But things, I think, have hit rock bottom for funding in Canada. I think things are starting to get a little bit better. Grant success rates were down to 14%. I think they're around 21% this last round. Doesn't sound like a lot, but it makes a huge difference. I think there's challenges for young men trying to get academic jobs right now, because there's a huge focus in trying to balance genders and ethnic backgrounds, which is fair and unfair. There's 2 sides to it. It depends if you overcompensate to adjust for past problems. I think we need to do a better job of educating the public in science and their awareness of science, because until the public starts to demand more funding for young scientists that are growing up, and more young kids get engaged in science from different ethnic backgrounds, we'll always have some sort of disparity in the system. So I guess what I say, it's a waste of time for scientists to approach politicians asking for money, because we're self-serving devils as far as they're concerned. It needs to be, the average person in the public that says they're a local politician. Look, why do we spend 80 times less than the United States on research? What is it that American politicians get that you don't get? Right?
Yeah. I mean, the funding situation in academia here in the U.S. isn't as much better than in Canada.
No, it's not.
But
the big problem is the fact that people have to derive a very large proportion of their salary from the NIH grants. They wouldn't need as many NIH grants, and the overheads their universities receive there, some of them are outrageous.
Yeah. I think one of the major differentiators between Canada and the U.S. is that industry jobs, the market is booming in the US. Living in Boston, Seattle, San Francisco area, the Bay Area, coming out of a PhD or a postdoc, sometimes they handpick you because you have that expertise and you can very easily, depending on your situation, on your background, transition into industry, whereas in Canada, that is dying out or non-existent, except perhaps in the Toronto area.
In the Toronto area, and I think, Eric, which, Michel Bouvier has had a big part of growing, spinning out some small pharmaceutical companies that will have potential in the future. The sad thing is the loss of, large companies like Merck and AstraZeneca,
Yeah.
Montreal Merck, because, the site in Montreal for Merck was extraordinarily successful over the years at developing new drugs. So, I guess, I'm a Canadian. I'm focused on the future of young Canadian scientists because that's what I'm training. I guess the decision is whether or not they want to stay in Canada or whether they want to continue working in the pharmaceutical industry in the United States. I know that many of my students who are looking to come back to Canada as faculty from the United States are finding it very difficult to find faculty positions. And a lot of that has to do with COVID and, everything's sort of shut down. So hopefully, some of our more senior scientists who are in their mid-70s will decide that maybe, retirement's not a bad idea. So we can open up those academic positions to the younger people that we're training. I know for a fact that the day I turned 65, I'm walking away. So.
Thank you for that. Two last questions. I think we've kind of talked about this, but any other top 3 aha moments that you had as a scientist that shaped your career trajectory?
I think the major one was my grandfather and his having Alzheimer's. I think if I had it all to do over again, I would become a cancer biologist like my sister. I think that's the disease is the biggest challenge for the future at the moment. Aha moment. It's hard to say. I guess just the fact that we've observed that some of these drugs actually do have benefit in animal models of disease. It's caused me to have to develop a huge amount of patience because from start to finish for each one of these projects, it's 2 years. From the time it takes to breed colonies of 16 mice per group, to aging them to the correct age, to then testing them with drug and then doing all the experiments. I guess the other major aha moment was when we decided to age male Alzheimer mice to 18 months of age and discovered they became just like females. They no longer responded to A-beta. They're no longer bound to A-beta. That's not saying that old men are like young women. That's not what I'm trying to say.
I still find the result absolutely fascinating and would love to know why is that so different for this A-beta 42 and mGluR5 interaction. It's just yeah. While you were explaining and you were talking through the story in my mind, I was like, okay, so why would that be different? Is it the receptor? Is it the environment? Is it the cellular components that might be different? And I thought about it, the sex hormones, but then you mentioned that you've also accounted for that. It's still, I think, a mystery, but very interesting one.
I think there has to be one of two things. There's either something that's missing in male mice that female mice have, or there's something missing in female mice that male mice have, or that this prion, we know if we take away the prion protein, we lose A-beta binding to the receptor as well. So maybe there's another member of that protein complex that is differentially regulated in some way between male and female mice. I don't know. I wish I had the answer.
Well, hopefully we'll get an answer at some point before you go full circle and you mentioned at 65 you would walk out the door.
Well, there's other things I'd like to do.
Fantastic. All right. Last but not least, if and when you have job openings in your team, where can people find you?
I don't really advertise, so you can find me by email, basically. My lab runs a Twitter account that I don't control. It's controlled by my lab manager. It's mostly just, to congratulate the successes of people in my lab and other labs. The best thing is to send an email. I can tell you that in Khaled's case, he didn't apply to my lab. His PhD supervisor applied for him. And I sort of, wow, this guy thinks a lot of this too. So, that's, a novel way of thinking about it.
And yet it worked out great because he's just phenomenal.
Well, I think Caron is one of my two most successful postdocs, the other one being Fabiola Ribeiro, who's back in Brazil. Who started all my Huntington work. So, yeah.
Fantastic. Well, I was going to mention the Twitter account. I did not know that you were not in control of the Twitter account, but people can tag you and then you'll definitely get the message or shoot you an email.
Okay, that'd be great.
All right. Thank you so much, Steve, for your time. I really enjoyed our conversation.
Thank you.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guest, as well as our team members, Attila Forrest and Ines Pinheiro. Please make sure that you subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, please leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget to check out and register for the Dr. GPCR ecosystem at drgpcr.com/ecosystem. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.